Hi guys, Miss Francis here to talk to you today about sex linkage. When we left off, we were talking about Mendel's patterns of inheritance, and around 1900, cytologists and geneticists began to see parallels between the behavior of chromosomes and the behavior of what Mendel called factors. Now remember, Mendel had no knowledge of chromosomes, of DNA, of alleles, but now scientists are beginning to get this technology and are investigating these features. So for example, these scientists discovered that chromosomes and genes are both present in pairs in diploid cells. They figured out that homologous chromosomes separate and that alleles segregate during meiosis. That follows the pattern of Mendel's law of segregation. And lastly, scientists learned that fertilization restores the paired condition for both chromosomes and genes. Take a look at this picture. What do you see? And take a look at this picture. What do you see? If you are not colorblind for red-green, you should see the letter 8 and the letter 5. Well, color blindness is an example of something that is does not follow Mendel's pattern of inheritance. So Mendel did not study anything that was sex-linked. He didn't study any traits that were sex-linked. Thomas Hunt Morgan was the scientist who actually discovered how genes can be linked to the sex chromosomes. And unlike Mendel, who worked with pea plants, Morgan worked with fruit flies. Why did he choose fruit flies? Similar to the reasons why we chose fruit flies for our lab. Fruit flies are prolific breeders. A woman can lay up to, not a woman, a female can lay up to 500 eggs at a time. They have a generation time of 2 weeks, and they only have 3 pairs of autosomes and a pair of sex chromosomes, XX in females, XY in males. So that makes it very easy to use fruit flies for genetic studies. And when Morgan was doing his studies on these fruit flies, he started to look for different variations, and he discovered a variation, what he called a mutant phenotype. Now, normally, fruit flies would have the wild type phenotype for eye color. So let me back up a little bit. Similar to how Mendel studied a trait that had 2 versions, for example, Mendel studied flower color, there was purple and white. Morgan studied the trait for eye color, where there's either red or white. So what he discovered was a male fly with white eyes, which was different from the wild type, red eyes. And what he did was similar to what Mendel did, where Mendel crossed a purple flower and a white flower. Well, Morgan crossed his white-eyed male with the red-eyed female. And the result was F1 offspring all having red eyes. Just like when Mendel did his cross of white and purple flowers, all of the offspring were purple. So this followed along with Mendel's pattern of inheritance, where the law of dominance comes into effect. So Morgan concluded, well, I guess the red allele for eye color must be dominant to the white allele. So what do you think Morgan did next? Exactly. Then he crossed his F1 flies. And what he expected was this 3-to-1 phenotypic ratio, where 3 of the flies would have red eyes and 1 fly would have white eyes. Just like in Mendel's flowers, 3 of the flowers were purple, 1 of the flowers was white. That was the 3-to-1 ratio in the F2 generation. So Morgan began to thought, okay, well, we've still got Mendel's pattern of inheritance. But Morgan discovered something else. He looked at the white-eyed trait, and he discovered that that trait appeared only in males. Now, all of the females had red eyes, and half of the males had red eyes, but the white eye appeared only in males, and the red eyes appeared only in females. So he figured, well, there must be something additional going on. And what Morgan discovered, the additional that was going on, was that a fly's eye color was linked to its sex, to whether it's a male or whether it's a female. And he called this a sex-linked gene. What that means is the gene that codes for the trait you're investigating is carried either on the X or the Y chromosome. A majority of sex-linked chromosomes are carried on the X chromosome, but there are traits that are called holandric traits. For example, a hairy pinna, which is hair on the earlobe, that's when the gene is carried on the Y chromosome. Just a caveat, the SRY gene, the gene that codes for gender, is also carried on the Y chromosome. But some other traits that are linked to the X chromosome are hemophilia, color blindness, Duchenne's muscular dystrophy, male patterned baldness. So going back to how we started, color blindness. If a son was colorblind, where did he get that gene from? Where did he get that trait? It had to have come from his mom, because the gene is carried on the X chromosome, and mom gives the son his X chromosome. Dad, in order to make it a son, to make it male, had to have given that son his Y chromosome. So other examples of traits linked to the X chromosome include hemophilia. Hemophilia is a recessive trait that involves the absence of one or more clotting flowers, which means that someone with hemophilia has prolonged bleeding, but it can be treated with intravenous injections of the missing protein. Duchenne muscular dystrophy. Unfortunately, people with this disorder rarely live past their early 20s due to the absence of an X-linked gene for a key muscle protein, which is characterized by progressive weakening of the muscles and the loss of coordination. So why are males more likely to get sex-linked traits that are linked on the X chromosome, than females? Well, because males only have one X chromosome. So if they get the X chromosome that's affected, they show that trait. Whereas females have two X chromosomes. So females can be carriers for the disorder, where males can't be chromosomes, right? So males only have one X chromosome. So any male receiving the recessive allele from his mother is going to express that trait. Next time, we'll take a look at what Punnett squares and pedigree charts with these sex-linked traits look like.